Okay, everybody, I'm going to, if John Kovliski could sit down because everyone's taken his talk, I've actually gave John one of my slides, which he kindly used as well. So I can sit down now. I'm going to talk to you about biocontrol, and primarily about calicivirus. And in that, I'm going to mainly focus on RHDV2 because I've got 6 minutes, so I'm going to be talking fast. And I need to acknowledge my co-authors here. I think Greg's in the room, and my Lindell's here, and others aren't here who've done a lot of this work. And to the Wedgie for giving us this tissue sample. Okay, so one of the first take-homes for you, I'm going to just cover off on a few things that you may or may not have heard about, and that is that RHDV2, the new one that's out there, is killing rabbits that are immunised against the old strain of RHDV1. We found this out at our Turretfield study site where we've been going to now since 1996. And this rabbit here is one of those that we published on. Had a very high titers of RHDV1. We'd caught it 7 times, and then when we did find it, when I found it dead, and we were able to publish this paper, and we're about to publish an update paper showing that it had died from RHDV2. So that's the first take home for you. RHDV2 is killing RHDV1 rabbits, so it's having a massive impact. This is another one that is Greg's, who's in the room, who can answer more on this than I can, and Greg's been taking our Turretfield data and looking at it in terms of myxomatosis. And what you see here between the grey bars and the yellow bars is that because of the timing of RHDV2, which is tending to come earlier in the year, we saw with RHDV1 that it came a bit earlier, started to come a bit earlier, but RHDV2 has come some weeks earlier. And what is happening now with myxomatosis is that we found myxomatosis is now in those yellow bars, coming primarily later in the year and really hammering the young rabbits. So that's another change that's happening, and Greg is about to, hopefully in the next few weeks, send our manuscript off to a journal. So myxo is having severe impacts, and more so on kittens. On the data that, another paper that Greg has led the authorship on our data from the Flinders Ranges, where I used to be the ranger back in the old days when I met Mark in '95, to our Turretfield data now, you see RHDV2 turning up where the red arrows are there, and the populations have declined significantly. And what Greg found when he analysed the data, the spotlight data from the Flinders and the trapping data from the Turretfield, is that both populations, one semi-arid and one temperate, have both declined about 80% compared to the 10-year average. So this RHDV2 is having broad impacts, hopefully, across the greater Australian range of the rabbit. This is the slide that I generously shared with my colleague John, and which you've already seen. And of course, the point here, which I won't dwell upon because you've already seen it, is that RHDV2, I asked Brian would he redo his 2010 graph, which he did kindly for us. He's predicted in the dashed lines there what actually did happen. But as you can see with RHDV2 now, with some of the more recent data just right at the end there, that the RHDV2 has driven the population down markedly so. And if we do follow the European situation, we would be expecting that to be sustained for quite some time. All right, Lindell, a co-author who's in the room, has been collating the usage of 1080 by NRM boards across the state. And what you can see here with RHDV2 coming in is that the use of 1080 for the control of rabbits has declined, and has declined dramatically. And we attribute this to RHDV2. Now, when Greg looked at the economic impact of what this means to landholders, it comes out to be about $1 million in 2017 from RHDV2's impact on saving landholders. Of course, this is a bit of a problem in that this is a time when you want landholders to be using more 1080 and getting rid of the residuals and keeping them down, as per John's showing you the Victoria Rabbit Buster campaign data. This is a paper that was published that Greg found, which was quite remarkable in that this is some satellite data, and when they found a break point in the veg cover, they found it came out to be around 1995, which happened to be when RHDV1 got out. And this is their pre-breakpoint data in orange and post-breakpoint data in the purple there. The paper's down the bottom if you want to look it up. But I found this to be quite amazing in showing that these guys are actually able to see in spectral analysis a change in vegetation cover, in this case from the Simpson-Strzelecki Dunefield region. Why this is so significant, and I think Reese Pedler might have presented on this at the last version of this conference, is that there has been significant large landscape scale recoveries of these 4 Australian animals. Some of them are threatened, and in the case of the Cresttail Mulgara, you see the red dot on the time is up. I'll be quiet. Red dot on the map there from the Sturton National Park. They're heading up there next week to trap again. That's another range extension for that species. So under RHDV2, maybe we're going to get even more ecological recovery. So that's what I'd be hoping for. Thanks for your time.